Okay, so apparently since people can't actually follow very simple instructions, then we'll try it again, just this time without any chat features available whatsoever. So obviously we've already gone through the P2 unit and we've already gone through half of the C2 unit. But for those of you that are going to be joining here, then we will go through some more of the C2 and some of the B2 as well. So this is obviously not got the chat enabled for the very simple fact that apparently people can't be trusted. So that's fine. We will not have the chat actually enabled. So that means you can't actually make any requests at this point. But it does mean that those people who want to learn can actually learn. And the rest of you can obviously go off into your own little existence now and do your own thing. So we've already gone through the main part of C2 in terms of the structure of the Earth. We've done P2. So now what we're going to carry on with is looking at the Haber process and how we make ammonia then. So as we said, we've got that very important symbol of reversible reactions, which is very important there. And in order to actually make ammonia, we need nitrogen and we need hydrogen. So what we actually find then is nitrogen plus hydrogen with our reversible reaction sign is going to make ammonia. Now, you do need to know the equation there for it. So we need to know the balance symbol equation, and that is for foundation and higher tier, which quite simply is going to be a case of, oops, I'll write it a bit clearer for you. So when you start off with this, you can have nitrogen, which is N2, hydrogen, which is H2, and then you've got NH3, which is your ammonia. So there are two molecules of ammonia, because obviously if you've got two nitrogens over here, you've got to have two nitrogens over there. And then because you've got 2 times 3 gives us 6, and there's two hydrogens there, then we've got to have a 3 in front of our hydrogen. So just remember, with the balancing, just start in logical place and then work it out. For this one, you could just learn it if you're not very good at balancing, because it's the only one that could ever come up for it. So make sure that you remember the reversible. In terms of the conditions we need for the Haber process, then we're going to have a high pressure. We've got a temperature of around 450 degrees Celsius and an ion catalyst. For those of you that really want to learn the pressure, then it'd be 200 atmospheres. Now, in order to reduce the costs, we also recycle any of the unreacted gases, because that obviously means we're spending less on the raw materials in the first place. Now, what we find is that they do like to give you a delightfully complex little graph. So when you're looking at a Haber process question, or even just a reversible reaction question, they do like to give you one of these graphs that has the pressure on the bottom on your x-axis, your yield on the y-axis, and then it's got a whole range of different lines that represent different temperatures. So you've got a couple of options here. If you're good at reading graphs, then you could just read the information from the graph itself. If you're not great at reading graphs, the good news is that the Haber process follows the same pattern every time, so you can just memorize it. So what we're looking at there is, as you increase the temperature, then you decrease the yield, because as you can see on there is, as the temperature actually increases, so it's getting higher, the lines are actually going lower, which means the yield is decreasing. But as you increase the pressure, then you increase the yield. So they're the two patterns to remember. Increasing the temperature decreases the yield, increasing the pressure increases the yield. And yield, quite simply, is just the amount of product that you make. Now, what we find for the higher tier is you need to understand why we use those conditions, okay? Now, the reason behind that is to give the lowest cost for the ammonia we're making at the end. So we use 200 atmospheres because obviously if you actually have a higher pressure, we have a higher yield. So that's good. But if we have too high a pressure, then in order to actually build the plant to withstand those pressures, it'll be ridiculously expensive. So 200 is the compromise. It's high enough to increase the yield, but not too high to cost too much to build the actual factory. In terms of the temperature, we know that the higher the temperature, the lower the actual yield. So you think, well, why are we using 450 degrees Celsius? And the simple reason behind that is that we need it to be hot enough to have a faster rate of reaction. So think into your C3 work there, that as you increase the temperature, you increase the rate of reaction because there are more frequent collisions. So it's, again, the compromise between having a faster rate of reaction, but not driving the backwards reaction too much. And we can actually also increase the rate of reaction by using an iron catalyst. So remember, the catalyst in terms of the Haber process is iron. Now, the next thing we're coming on to is looking at making salts. First part of this is we need to remember, going back to, again, year 7 work, what's an acid, what's neutral, and what's an alkali. So if you're thinking about an acid, it's pH 1 to 6, and using universal indicator, they're the reds, the oranges, and the yellows. If you're thinking about the alkalis, they're the 8s to 14s, and they're blues and purples, and neutral is pH 7, which is green. If it asks you the pH of something, then do not write a colour. Okay, if it asks you the pH, it wants a number. So acids 1 to 6, neutral 7, and alkalis 8 to 14. Now, the reason that we've got those different pHs is all down to the concentration of the hydrogen ions. So the greater the concentration of hydrogen ions, the more acidic it is. Okay, so that means it's got a lower pH. If you want to know whether something's acid, alkali, or neutral, then what we actually have is an indicator. So these are usually things like plant dyes that change colour in different pHs. Litmus is one of the common ones they like to ask you about. It will go red in acid or blue in an alkali, but it doesn't show you how acid or alkali it is, just that it is one. You can take one step better by using universal indicator, which obviously shows you the range of colours for the different pHs, but it's not always easy to match those colours. If you've used the universal indicator, then it's quite hard to match it on that little scale. So the best way, or the way you can improve that, is by using a pH meter. So it's just a little digital probe you put in, push a little button, and it gives you a reading of like pH 7.3, and then you've just got it instantly. If we're looking at neutralization, that's where we're reacting an acid and a base. Now we make two things, we make a salt and we make water. Now, what we need to understand is the name of the salt, okay? So there are 4 different endings we need to remember here based on the acids that we could use. If we're using hydrochloric acid, it makes a chloride, sulfuric acid makes a sulfate, nitric acid makes a nitrate, and phosphoric acid makes a phosphate. So you've obviously got those different acids, and that makes the ending of the salt name. The first part of the salt name is always just the name of the metal. So quite simply, when you look at the alkali, look and see what the first word is, write that down, then look at the acid, and if it says sulfuric, write the word sulfate at the end. And then all you got to do is plus water. The only exception to that is if we have a carbonate, and then it's going to be our salt plus water plus carbon dioxide. So don't forget, carbonates also make carbon dioxide. The one little tricky one they like to throw in, particularly for those of you on the higher tier, is talking about ammonia or ammonium carbonate. And when you're writing the name of that salt, you need to write it's an ammonium, so IUM, and then its ending. Okay. So higher tier, as I said, you may have to do a balanced equation, but I'm not going to go through balancing tonight because I'll be honest, if you don't know how to balance it now, chances of you learning by tomorrow isn't so great. But do remember, balancing equations, you get 2 marks. 1 mark if you write the formulas, and the 2nd mark for the balancing. So even if you can't balance it, write the formulas down because you'll get half your marks. Now, one of the things that we will actually be doing through making these salts and so on is making our fertilizers. So when we're thinking about fertilizers, they're all designed to make plants grow faster and to make them grow bigger. Now, the reason they do that is they've got 3 essential elements. Now, essential elements are basically elements that plants need to grow. So you've got nitrogen, phosphorus, and potassium, so N, P, and K. Now, nitrogen, phosphorus, and potassium are very important to our plants, and this is also the question you're going to find one of their favourites, asking about the different number of atoms or the different number of elements present within a formula. So they will give you some random chemical formula, and they will say how many different elements are present within there. Count how many different capital letters there are, that gives you your different elements. If it asks you how many different atoms there are, then all you need to do is count up all of the individual atoms. So remember that where you've actually got ones where it's going to have, let me just write an example for you. So if you've got one like this, so NH4NO3, then you've got 1 nitrogen here, 4 hydrogens, another nitrogen there, and 3 oxygens. So you'd have 3, 4, 8, 9 different atoms. But in terms of the elements, obviously we've got nitrogen, hydrogen, and oxygen. So you've only got 3 different elements. So just to go careful on that one to make sure that you get it right, because those should be a couple of nice, easy marks. They could also ask you here about how plants actually get fertilizers. They absorb them through their roots. Okay, so it's always the answer. And if they ask you why they've got to be soluble, it's because they need to dissolve in water to be absorbed through the plant's roots. Now, what we find is there are some big problems of using fertilizers. And the first one that we need to remember is this name, eutrophication. Now, basically what that means is that the fertilizers wash into the actual rivers. That means that we then have this algal bloom, so that's where the algae grows really quickly. That stops the plants getting their light in the actual water itself. Therefore, they die. And as a result of them being decayed by the bacteria, all the oxygen in the water is used up, and that means the fish die. Okay, so fertilizers wash into or run off into lakes and rivers. Then we get increased amounts of the nitrates and the phosphates in the water, and algal bloom occurs, plants are shaded, plants die, bacteria will decay the plants, fish die. Okay, so that's pretty much your steps for eutrophication. Now, what we actually find, one of their favorite questions for some reason, is naming a piece of equipment. Now, they are relying on the fact that you actually had a science teacher that did this experiment with you in year 10, because they're going to give you an actual diagram of the equipment and expect you to be able to name it. So what they're going to ask you to actually identify is this big thing here. So you'll have a clamp stand and you're going to have this big tube here. Now that is called a burette. Okay. Now the burette is what we're actually going to put our acid into to neutralize the alkali when we make a fertilizer. Okay. So just remember the word burette, because generally they say, what's the name of equipment X? And that will be your answer. So we're now coming on to the last little bit here, which is looking at salt. So first of all, we need to know where we get salt from. Now, what we actually have in England are quite a large amount of salt deposits underground. So what we have then is we can actually get it from underground through a process called solution mining. So all we do there is you just pump fresh water into the ground, it'll dissolve the salt, and then you pump the brine, which is the concentrated salt solution, back to the surface. Now, that's a continuous process. It doesn't need many people, so low labor costs, and we can have one very significant downside of it causing this thing called subsidence. So that basically means that you've eroded all of the ground underneath the house so it can sink into the ground. Now, once we've actually got that brine, we generally want to split it into more useful things. So we use electrolysis here. So what we're going to do is use electrolysis, and that's going to split the brine up into 3 chemicals. It will split into hydrogen, chlorine, and sodium hydroxide solution. The hydrogen itself we're going to use to make margarine, because what that's going to do is allow us to actually spread that without it running everywhere and having the same problems with all the butters. Chlorine we use to sterilize water. Those of you doing C4 in next week will hopefully remember that. And sodium hydroxide is used to actually make soap. Okay, so just remember those 3 uses. First of all, hydrogen for margarine, chlorine for sterilizing water, and sodium hydroxide to make soap. Now, the way we actually split this up is in good old electrolysis again. Now, electrolysis works in exactly the same way as the copper. We still have the anode and the cathode. So you've got the positive and the negative. The only difference this time is what's going to be forming. So you pass the electricity through your brine solution. And what we're going to find is at the anode, which is the positive electrode, you get chlorine gas because chlorine is negative. At the cathode, which is the negative electrode, you get hydrogen because hydrogen is positive. And then coming off the bottom, we'll just literally run off the sodium hydroxide solution. So that's literally what we have happening in terms of our electrolysis of brine. The only other thing they could ask you on the chemistry paper is how we test for chlorine. And quite simply, all you're going to do is get some moist blue litmus paper and it turns red, then white. So you always get a mark for saying the moist blue litmus paper turns red. That's worth your first mark, and then turns white for the second. So that is all of our C2. So the last thing we're going to go through is a bit of chemistry, not chemistry, biology. we're not going to go through all of the biology because that would take a very long time. So all I'm going to do is I'm going to pick out those few bits that people have asked to go through earlier on today, and the rest of it, obviously, is on the channel if you want to watch the videos. So first of all, the binomial naming system. Now, binomial just means two names, okay? Bi meaning two, nomial naming. So all we do in science is every single living thing on the planet has this binomial name, and it's normally in Latin. Now, the reason for this is that it doesn't matter what language scientists then speak, they are talking about the same organism, so it avoids confusion. So they will always have the same two-part name. The first part is made up of the genus, and the second part is the species. Okay, so if they ask you a question where they've given you an example and a load of text at the beginning and ask you to fill out a table with all the different classifications, so the kingdom, phylum, etc., then all you need to do to get either the genus or the species, whichever one they've missed out, is look at the bit they've written in italics, and if it's the genus they're asking, it's the first word. If it's the species that's missing, it's the second word. So just pick those out. They do like to ask you to define the word species as well, and quite simply, that could be a 2-mark question. It's a group of similar organisms which are capable of interbreeding to produce fertile offspring. So you get 1 mark for saying a group of organisms which are capable of interbreeding. There's one. Second one, to produce the fertile offspring. So make sure you do remember that side. They love to ask you about adaptation, okay? I'm not going to go through adaptations of different animals because most of it's quite a lot of common sense, in all honesty. If it asks you about adaptations of a polar bear or whatever animal, it gives you a picture of it, think about how it's adapted to survive and then just write about those. Now, we did have quite a few requests for different cycles in terms of our B2. So first of all, we have the carbon cycle. So you have one of my amazing little diagrams. So the carbon cycle then, we're going to start off with carbon dioxide in the air. So with carbon dioxide in the air can only be removed from the atmosphere through one process, which is photosynthesis. So what they tend to do is they'll give you a simplified diagram with some boxes and ask you to fill out the actual missing words. Now, the line that's going to be pointing from carbon dioxide in the air to a plant, you're going to write photosynthesis for. If you've got an arrow going from the plant to an animal, that's a sheep in case you couldn't tell, then that is going to be feeding or eating, okay? Any living thing, so animals or plants, back to carbon dioxide will be respiration. And then finally, the only other process they would ask you to name is combustion. So just look for anything like a factory, a house, a car, anything that's burning a fossil fuel, then combustion returns that carbon dioxide to the atmosphere. So respiration and combustion will be arrows that go back to the atmosphere and the carbon dioxide, whereas photosynthesis is the only one going from carbon dioxide to a plant, and then the arrow that goes from the plant to the animal, that's just going to be the feeding or the eating. So make sure you remember those key processes. It could be a 6-mark question, but even if it is, all you got to do is put those in sentences because it's not requiring huge amounts more than that. What we then come on to is the nitrogen cycle. So our second cycle is nitrogen. Now, this one is very different depending on whether you are doing foundation or higher tier. So you both need to know that we need nitrogen in order to make DNA and amino acids, and those are used to make proteins. So if it asks you, why do we use nitrogen to make proteins or to make DNA, there are 2 possible answers for you. But the only way that we can actually use nitrogen is when plants take them up as nitrates, okay? So we can't use nitrogen gas from the atmosphere directly. If you're doing the foundation paper, then you don't need to know everything on here at all, okay? So for those of you doing the foundation paper, you literally need to know that nitrogen in the air, okay, so the nitrogen gas is actually going to be taken in by plants, okay, as nitrates. Then the plants get eaten by animals, and then the animals die, decay, and back to nitrates. That's it for foundation. For those of you doing the higher tier, you need a little bit more detail because we've got some different types of bacteria involved here that we need to remember. 4 types of bacteria, in fact. So if we start off with nitrogen in the air there, then we've got the nitrogen fixing bacteria, first of all, that will take the nitrogen in the air and change it into nitrates. We also have nitrates being changed back into nitrogen gas by the denitrifying bacteria. So those ones go in a little loop. We also have our nitrates then being absorbed through the roots of our plants. Plants then use it to actually build proteins. Those proteins are eaten by animals. And then what we find is when plants and animals die, they obviously get broken down by the decomposers. And decomposers are bacteria or fungi. Now, when they break down the proteins, it makes ammonia. And then the nitrifying bacteria will change ammonia into nitrates. And then we go back round the little loop once more. So 4 types of bacteria to remember. Nitrogen into nitrates is the nitrogen fixing. Nitrates into nitrogen denitrifying. Ammonia into nitrates is our nitrifying bacteria. And then the decomposers take proteins and change them into ammonia. So you need to remember those 4 different types of bacteria and what they actually do. But if you can only remember a few of them, don't think you're not going to get any marks on that question. Still write about the processes and you will still get several marks on those questions. Now, the only other ways that we can actually get nitrogen gas back into the soil as nitrates, lightning, or obviously using fertilizers, other than obviously the nitrogen fixing bacteria. So we do have different ways of getting nitrates into the soil. So we've now covered nitrogen cycle and the carbon cycle. The only other bit that people wanted to go through was natural selection. So obviously we know that there are different adaptations that organisms have that allow them to survive better. Now, what we find is that Charles Darwin many years ago came up with this whole theory of natural selection to explain the process of evolution. So first of all, he noticed the fact there were very large populations. So what we have are lots of organisms being born, but not all of them are surviving. So that led to this idea of survival of the fittest. So what we actually have in terms of our natural selection is we start off with a large population, which has variation within it. Okay, so make sure you mention variation. That means that some are more adapted to surviving in those conditions than others. So they will survive. They will then reproduce and pass on their DNA or their genes to the next generation. And over time, that leads to evolution. So remember, we have variation in the population. Those that are best adapted to the environment will survive. Those that survive reproduce. They pass on their genes or their DNA. Don't use other random phrases, okay? Don't say they pass on their features or their characteristics. Say they pass on their genes or their DNA. So that's our evolution. We do have, obviously, for the higher tier, that idea of good old Jean Lamarck, who was that French biologist. Now, he was the one that decided that giraffes really quite wanted to eat those leaves high up on the tree, so they stretched their necks as high as they could, and then they passed those longer necks on to their offspring. Obviously, now when we look back on it, we think, God, what an idiot. But obviously, he didn't know about DNA in those times. So his idea, while it sounds utterly ridiculous today, explained it at the time, okay? But since Darwin, obviously, we have natural selection, which explains it much more accurately. We do have a few examples of evolution in our everyday life. The peppered moth is one. So you have the light-coloured and the dark-coloured ones, all about camouflage. So pre-industrial revolution, the dark-coloured moths used to stand out, and therefore there are fewer of them in the population. Post-industrial revolution, everything was rather dirty, so that meant the light-coloured moths stood out more, so they got eaten, and therefore the population shifted. You've got antibiotic resistance, which is obviously where some bacteria are resistant to antibiotics that we use, hence why you should take your full course of antibiotics when it's prescribed from the doctor. And we've also got the rat poison warfarin, which is now obviously a problem because our rats are becoming resistant to that too. The only other thing I want to go through with you tonight before we finish is talking about conservation. So in the world today, what we have are many species that are actually endangered because of basically us as humans, let's be honest. So what we find is that if we don't do something about these endangered species, and therefore they fall beneath the critical level, which is the number they need in order to survive, then they will become extinct like many other organisms over time. So what we find is that we can actually try to solve this. First of all, if we protect habitats, we can obviously ban hunting or trade of various parts of them. We can educate people. You can carry out captive breeding programs in zoos. anything like this is going to hopefully help to improve the numbers and therefore stop them becoming extinct. So do remember that there are obviously certain topics that come up time and time again on those papers. So I've gone through as many of them as I can, but for a quick recap on those topics that I would look over before your exam, if you still have some time and a bit of energy, then I went back through all the papers over the entire course of the current Gateway Science course. And for the higher tier, the questions you want to look at in B2, particularly evolution and natural selection, that's come up on 5 different papers now. You need to know about adaptations and conservation, as we've gone through the nitrogen cycle, which we've already gone through. Make sure that you know that classification system, so kingdom, phylum, class, etc. Make sure you know those in order. That comes up quite frequently. Obviously understand about energy transfer in food chains on the higher tier, the fact that food chains are limited in size because we do lose energy at each stage by excretions, egestion, etc. And therefore they're limited in their size because eventually not enough energy gets passed on. For the chemistry on the higher tier, make sure you can name the salts, make sure you know about fertilizers and how they're made, make sure you know the Haber process and reversible reactions and how to read those graphs, and electrolysis has come up 4 different times. So make sure you know those key bits as well. Lots of other things have come up, just not in the same number of papers as those ones. And then finally in P2, alpha, beta and gamma radiation has come up in virtually every paper, as has the energy efficiency calculation, the cost of electricity calculation, redshift and generating electricity. If you're doing the foundation tier paper, then classification, again, that comes up. Adaptations and evolution, again, very common. And obviously things like the predator and prey and their adaptations as well. Make sure you go over the key things about predators and prey. On the chemistry section, they're looking at fertilizers again, reversible reactions, acids and alkalis and alloys come up more on the foundation along with the salt names. And then finally in the physics section, still alpha, beta and gamma, still the efficiency calculations, generating electricity using solar panels and wind turbines this time, manned and unmanned space travel, asteroids and their differences, and then the theories on the universe and also how we actually travel in space, as well as the order of the planets. So make sure you know the calculations. And the only other thing that you need to bear in mind is that you have section D at the end, 10 marks just for reading the question and obviously interpreting the information they give you. So that's it for tonight. I'm sorry that we couldn't keep the chat going, but obviously it wasn't really the most beneficial thing to have going. So for those of you that are doing additional science this year as well, we will run live seminars for the Unit 3s and the Unit 4s on the night before, and we won't have chat going on those. So if you want to go through any key topics, then you have to post a comment onto the actual website beforehand, and I will make sure I add them to my list to go through them. Otherwise, good luck for tomorrow, everyone. Make sure you don't stay up all night revising. Do try and go to sleep, but make sure you put a calculator in your bag, because we all know physics will have at least one calculation worth 2 marks tomorrow. So good luck, everyone, and I'll see some of you again next week.